Hello everyone, I'm Brighton Wajila and it's a pleasure to welcome you to our exciting session of our CIG weekly reading group. I co-host these sessions with my colleague Mkenya Lukman, joined by Yasa Mamam from Florida International University in Miami. Yasa will be sharing his groundbreaking research titled Insights into the DNA and RNA Interactions of Human Topoisomerases Simulations. This is a phenomenal opportunity to gain a deeper understanding of this topic from an expert. If you are interested in more sessions like this, don't forget to check out the details in the description below. Let's get started.
Insights into the DNA and RNA interactions of human topoisomerase 3 beta using MD simulations. So I'm going to talk about the specific protein, the enzyme human topoisomerase 3 beta, and how it interacts with DNA and RNA. So going forward, I'm from the biochemistry PhD program in FIU. So FIU is Florida International University. It is located in the state of Florida, like at the very southwest edge of the United States, in the city of Miami. So we are not exactly located in the city of Miami, but a bit west in a suburb of Sweetwater. Miami, I guess you guys heard about Miami. It's pretty famous for its beaches and nightlife. And this is like a short picture of our university. And I'm in the biochemistry PhD program under Dr. Prem Chapagin and Dr. Yuking Shedin. So this is a combination of both of their expertise. This project, Dr. Shedin is a pioneer in type 1A topoisomerase research, and Dr. Chapagin is an expert in molecular modeling and molecular dynamic simulation. So I work under the supervision of both of them to study the structures of, sorry about that, structure of type 1A topoisomerase, and specifically human topoisomerase 3 beta, and how it interacts with DNA and RNA. So as you know, DNA are very large molecules. They have to be stored in a very small space. And how big is this DNA? So if we stretch the chromosomes in our body, they can be up to 2 meters, which is pretty long, right? And they have to be stored in a very condensed way, but they also have to be pretty well stored because cellular machineries have to reach them. Okay, so I found this interesting fact that if each base pair was 1 millimeter apart, which they're not, but let's say if they're 1 millimeter apart, so the gene in one cell, okay, so our chromosomes would be about 3,200 kilometers and stretch across one end of the continental Africa to the other end, going right through Uganda, your country, right? So wow. Only 300, so, and not all of it is translated to RNA. So if our chromosome was stretched to 3,200 kilometers, every protein producing DNA would be every 300 meters, right? So not all of it is translated. So you can understand that it needs to be stored in a nice way where chromosomal machinery can reach it. But this is not always easy. Because let's say a small DNA like plasmid, right? It is a closed system, and larger genetic DNAs are also, they're so large, they can also be considered closed because they're so large and they cannot rotate that easily. The edges are closed. And this is where supercoiling in DNA comes in. So the picture on the left, like we don't really see these anymore, but if you guys have seen them, right? So the phones used to be like these. They used to have wires and cables, and these cables would wrap around itself, right? So first the cable itself is coiled, and then it wraps around itself. This is known as supercoiling, and this exact phenomenon takes place in DNA. So DNAs are double helices, right? And they wrap around, it wraps around itself along the axis. And then this whole, let's say this is a plasmid DNA, which is circular, it will wrap around itself, leading to DNA supercoiling. And this kind of supercoiling is like a very highly researched subject, and this is a matter of great discussion among even mathematicians. The twists and the rides and the linking numbers of DNA supercoiling, which is a subject in itself, and it is really like a very big and deep subject. So I'm not going to go too much into it. But as you can understand, if DNA is supercoiled like this, it needs to be opened up for daily cellular activities like replication and translation to take place, right? So let's say translation is taking place, the RNA polymerase is going across the DNA and reading the bases ACDT. It has to open up. During replication, the helicase also moves along this super DNA double helix, and it has to open up for the new DNA to be made. And as you can understand, when you open up a coil like this, it makes even more coils downstream, right? And this leads to so much supercoiling, the cell can become unstable. So what kind of topological barriers do we usually face in the DNA, right? So for one thing, I said replication. The replication machinery is moving forward, the helicase is opening up the DNA, and there is positive supercoilings formed in front of that helicase, and there is precatenance that needs to be separated from each other for the new daughter chromosome to take shape. And during transcription, the RNA polymerase moves forward. This is known as the twin coiling domain model. So there is positive supercoilings forming in front of the RNA polymerase and negative supercoilings that form behind. Other than that, once two new chromosomes are made, they have to be separated from each other, hemicatenance, and they can like tie up themselves in such a way that also has to be relaxed. So these are known as DNA knots. So as you can understand, this is a very knotty subject, and these things need to be taken care of. Otherwise, the cell would die because the cellular machineries won't be able to reach those places. So this is where DNA topoisomerases come in. And this is what they do. On the left-hand side, you can see this is a small GIF where the topoisomerase is binding to a double-strand DNA, and it's a supercoiled DNA. It can bind, it can cut open, it can pass another strand of DNA, and then reseal that cleaved DNA, right? As if by magic. And they are aptly named the magicians of the cell because they can do this. They can do this without harming the DNA. They can cut it, pass a strand, and relegate it. So let's say just a simple plasmid DNA on the right-hand side in E. coli. So you can understand that negative and positive supercoilings are taking place, and topoisomerases like topoisomerase 1 and topoisomerase 2, the one in this small animation is topo2. It can cut double-strand DNA and pass double-strand DNA, and there is also other variations that I'll just show in the next slide. So they can work on single strand, so they can bind to a single strand, cut and pass it, thus relaxing the DNA. This is a topo1 that's found in E. coli. And there is the topo2, which works with the help of ATP. It binds to a double strand. It can relax the supercoiled DNA. It can decatenate two DNAs. It can also induce relaxation, supercoiling. So topo2 can either reduce or induce supercoiling, and it needs an ATP to work. So there are several types of topoisomerases. First found in 1971, they have been regularly found in different organisms, and they have been studied extensively. The most basic types are the type 1, and well, the basic general classification is type 1 and type 2. Where type 1 works on a single-stranded DNA and the type 2 works on a double-stranded DNA. On the left-hand side, I have shown some examples. This is a type 1a topoisomerase, and all type 1a topoisomerases have this same shape. They have this toroid hole, which we think can open and pass DNA through a strand passage mechanism. It binds to a single strand, it opens up, and then the other strand passes through it, and then it relegates it. This is the type 1A, and this is found in all living organisms, right? They can be found in eukaryote, prokaryote, and everything. The next is type 1B. Type 1C is pretty newly found, and they work in a different way, is that they do bind and cleave a single strand, but they don't do the strand passage thing. They rotate around the intact strand and then relegate it. So this one is pretty unique in the sense that it is found in mostly eukaryotes and some other organisms, but not in bacteria, not really seen in bacteria. Type 2, again, different types. So in bacteria, they are mostly called DNA gyrases, and a very good and very important target of antibiotics like fluoroquinolones. And also in humans, they have we have type 2 alpha and type 2 beta. And they can work in very many ways. They can work on the relaxed to make it supercoiled, and they can work on the supercoiled to make it relaxed, all type 2 topoisomerases. And type 1 topoisomerase also can catenate and decatenate and so on and so forth. Again, I want to remind you the type 2 topoisomerases need ATP and the type 1 topoisomerases do not need them, but all of them need a divalent cation like magnesium to impart their activity. If you completely remove the magnesium, they can still cut the DNA, but they won't be able to complete their catalytic cycle. So it's just that they won't be able to relax the supercoiling. Again, I wanted to show you a few more things that goes on the human cell. So this is just the human cell and what type of topoisomerases take part in activities in the human cell. I wanted to focus on this part at the bottom. So when relaxation is taking place, when transcription is taking place, top3 beta, the enzyme that I'm going to talk about today, it works in this part, top3B, where once the transcription has taken place, the newly formed RNA can sometimes bind with the DNA and form a hybrid called R-loop. Top3 beta can work on that, and top3 beta are known to work on RNA knots that may be forming and RNA catenates to decatenate the RNA. So as you can understand, this is what makes top3 beta unique. In human cells, top3 beta is the only enzyme known that can, only topoisomerase that we know that can work on both DNA, single-strand DNA, and RNA. None of the others work on DNA. So there's this paralog top3 alpha. It has been shown only to work on DNA. They look almost the same, but there are some structural differences. But yes, top3 beta is the only one that one can work on RNA. And this is very important for our research, and this is what we focused on. Just to tell you guys a little bit more about the mechanism of topoisomerases, as you can understand, they have to cleave the DNA, right, to pass and relax it. So what they do is all topoisomerases have an active tyrosine, a toprim domain where the active tyrosine is. This active tyrosine does a nucleophilic attack on the DNA backbone and forms a phosphotyrosine bond. And it can either make a 5' adduct in the case of type 1A topoisomerases and type 2 topoisomerases. The other one, the swivel, is like the one that I said kind of swivels around the intact one. That one forms a 3' adduct, okay? So on the right-hand side, you can see like a small figure. Let's say if this is the enzyme, it has the tyrosine. This is a type 1A, example of type 1A topoisomerase. It has the active tyrosine. The active tyrosine can form a bond and break the DNA backbone. This strand will then pass through and it will relegate it, thus relaxing the supercoiling. Now, the two types of topoisomerases, type 1 and type 2, they have different pathway of imparting their activity. The type 1A and type 1B are also different. I'm just showing two examples here. One is type 1A on the right-hand side and the type 2 on the left-hand side. So as you can understand from the small animation that I showed before, the intact DNA strand comes in and through the T gate to the N gate, and then one of the segments, the G segment, is broken apart with the help of ATP. The intact strand then passes, the ATP is turned to ADP and released, and then finally the cleaved strand is religated and then passed through the end gate. Thus, this is the total catalytic activity of catalytic cycle of the type 2 topoisomerase. On the right-hand side, this has been a much more a matter of interest. Dr. Shedin's lab also does extensive research in the mechanism of and the catalytic cycle of type 1a topoisomerases. The current hypothesis or the proposal is that the C-terminal tail binds with single-strand DNA, especially in the negatively supercoiled domain. So the negative supercoiling induces a separation in the double-strand DNA, resulting in a single strand, and the C-terminal tail of the topoisomerase can bind with the single strand. The other one is bound with the active tyrosine, and there is a large conformational change where the gate opens along with the cleaved part of the DNA, and the intact strand is then passed through. The gate comes down again and it religates the cleaved strand. Thus, the linking number is increased by 1 of the negative supercoiling DNA and causing relaxation. So the matter, like if the way in this gate opens is still under investigation, and molecular dynamic simulation has been used to study it to some extent, but it has not been fully conclusive. So there is still research going on regarding the hinge that allows this gate to open and the other conformational changes that take place. So my study is not exactly based on this, it's a bit different. So why are topoisomers important? As you can understand, they can create double strand breaks, right? So if we don't answer that double strand break, we do not religate it, this will cause a lot of problems. The double strand breaks will accumulate and the cells will die. So you have to make sure that topoisomeres work precisely as they are intended to. And this makes them a good target for anti-cancer and antibiotic drugs. For antibiotics, because bacteria has DNA gyrase, we do not. Fluoroquinolone like ciprofloxacin are used to arrest those double strand breaks and lead to bacterial cell death. In human, top1A and top1B both, top2 and top1B are both targeted. Top1B is targeted through drugs such as candotecan, and top2 is targeted through irinotecan, topotecan. They can arrest those cell breaks, and because cancer cells have much more topo activity, they can arrest those double strand breaks, leading to the cell death of the cancer cells. So they are important drug targets. So as I have told you in the previous slides, the RNA activity is the thing that we are interested in, HTOP3 beta, because they are unique in that they work on both single-strand DNA and RNA. So during, like when they first found about topoisomerases, they could understand that if this topoisomerase works on single-strand, there is a very good chance it works on RNA. And in bacteria also they have RNA, right? But they have only one type 1a topoisomerase, so it makes sense that they are going to somehow be involved in that. And this was shown first that they can cleave, so they can just cut the RNA. Was first shown by Doctors Daiget and Marians in 1992. And in 1996, they showed with like a very simple but elegant experiment. So they gave the topoisomerase circular DNA, and it could make it a nice trifoil knot, so that it means that it could cut, pass, and relegate that RNA. So that's when it was proved that E. coli topo3, a type 1a topoisomerase, it has RNA activity and it can work in the cell. And the RNA activity of human topoisomerase 3 beta, as you can see from these crystal structures, they are almost exactly the same. This one has the DNA, this one does not. But it was shown in 2013 that HTOP3 beta has RNA activity by two separate groups of RNA. And as you can see, although they have the same N-terminal domain. So this is the N-terminal domain. This has been crystallographically resolved. They are not exactly the same. The H-top3 beta has a longer tail that has not crystallographically resolved, but E. coli topo 3 is pretty small. It has very short tail. It's almost entirely in this picture. And I'm going to talk about a little bit more about the N-terminal tail in the next slide. So as you can understand, the prokaryote and eukaryotic organism both have this type 1 topoisomerase, right? The one that works on RNA. But in bacteria, E. coli topo 3 and E. coli topo 1, they can both work on RNA. And their sizes are different across organisms. For example, in Archaea, these are pretty small. The E. coli topo 3 is also pretty small. It doesn't have a C-terminal tail, but still, all of them show RNA activity, right? In human, the H-top3 beta has a pretty similar N-terminal domain, which has that toroid shape like the E. coli ones, but it also has a long tail. And other eukaryotes, they also have this long tail and this RGG binding domain, which is observed in other RNA enzymes, RNA binding enzymes. And this C-terminal tail has been shown to be pretty important for its RNA activity. If this tail is cut off, it reduces its RNA activity. Interestingly, human top3 alpha, an isoform or parallel of the H-top3 beta, it also has a long tail, but it doesn't show any RNA activity, right? And yet in E. coli, the one that has a very short tail actually it barely has any tail. It also shows RNA activity. So why? Why is this happening? Maybe the N-terminal domain plays an important part, right? And H-top3 beta is also important in humans because they interact with another a protein. So it works in tandem with another protein. It's called TDRD3. It brings it in, and together they can work on the R loops that form during translation, and that has to be resolved. Otherwise, it will lead to cellular instability. They have been found pretty important in neurodevelopment. They have been shown to be pretty important in neurodevelopment in Drosophila. There have been patients found with mental retardation who lack the H-top3 beta, and they show very important interaction with mRNA. So most of their activity is mRNA related. Interestingly, deleting this is not does not hamper cell viability. So if you kind of knock out H-top3 beta, the organism won't die, but it will suffer shorter lifetime. So their whole lifetime is like affected with top3 beta knockout. And that's why we want to study H-top3 beta and why we do molecular modeling is that, well, for one thing, as you can understand, there are two things happening, right? It has to bind with the DNA and then it has to cleave it. So for cleaving, we need the DNA covalent complex where the topoisomerase is bound with the DNA, the attack has taken place, and there is a phosphotyrosine bond showing. But there is right now only one crystal structure of that. And molecular modeling, computational modeling can help us overcome that. We can generate those covalent complexes without a crystal structure, and we can compare it with the current findings and we can establish our results. Also, the H-top3 beta crystal structure that is now available, it's APO. It doesn't have any either DNA or RNA, and we don't really know if it was RNA there, how it would be different. So we try to model it with RNA. And magnesium, although it plays an important role, most of the crystal structures do not have that. Well, there is one MTB topo structure where there is the DNA and the magnesium in the binding catalytic site, or the binding site. And in H-top3 beta crystal structure, although the magnesium is there, there is no DNA. So we don't really know if the DNA was there, what we would see. So for all these, we kind of decided to model the H-top3 beta with the DNA and with RNA, and then covalent and non-covalent, which I'll tell you guys about shortly. And we can observe the residue and nucleotide interactions in a dynamic way. And finally, these models have further purposes. You can use them for different purposes later once you have established that your results are correct. So I wanted to hold this off for a little bit later because this goes directly with my experimentation. So now I'm going to talk about a little bit more about the structure. So I have shown you the 5GVC structure before, right? The crystal structure, but it doesn't have this tail where there is some zinc fingers and the RGG binding domain. So we are thinking that these zinc fingers are responsible for binding to the nucleic acid. But more importantly, we think that this RGG is responsible for binding to RNA as opposed to just binding to DNA. Now, this is an AlphaFold prediction. AlphaFold is like the hot top of town right now. They got the Nobel, and you can just use this to find structure. Now, it really predicts well the N-terminal domain, the same torus shape and everything, the top rim domain on all the domains. And it kind of also predicts the zinc finger, but there is some poor confidence in the loops and the very end of the tail. But it gives you an idea how it would look like if you could resolve it. Currently we have the full structure of the E. coli resolved crystallographically, but none of the humans. So as I have said before, this is the N-terminal domain, and the C-terminal domain is not conserved and very hard to find crystallographically. The other thing is that the C-terminal domain, although is very important, you can make a truncated enzyme. You can express a truncated enzyme, and it won't really lose the RNA cleavage activity, but it would lose a lot of the whole catalytic cycle activity. So it won't be able to religate it. And finally, there is the top rim domain that essentially holds a very important motif called the DXD motif, where there are two acetic acids that interact with the magnesium, and they're very important for the whole activity of the topoisomerase. If they're mutated or deleted, the topoisomerase won't show its activity. This is important because this also shows up in my computational models. Now, there is only one crystal structure available of the H-3 beta, and there is some more physiological study of topoisomerases, but not too many like core biochemical activity of how they interact with the nucleic acid. But this paper, which came out in 2022, it's a very good paper on H-3 beta. They cover a lot of ground on how H-3b interacts with DNA and how it imparts its activity. So some of the things they were able to show that if they give a 40-mer oligo, H-3b can cut it in specific places, specifically for the DNA. But for RNA, they show a lot more cleavage sites. And in DNA, they also show some sequence preference. It means that if the DNA cuts at one place, what would be the nearby bases be? So most of the bases are mostly thymine and cytosine at -5 position. So let's say if it cuts in this place, the topoisomerase cuts the DNA in this place, and these are the 5 primes, so -1, -2, -3, -4, -5. So there is a cytosine 5 bases towards the 5 prime end that is preferred, and there is always a thymine preferred at -2 position towards the 5 prime end, and +2 and +3 positions towards the 3 prime end. So 2 bases and 3 bases towards the 3 prime end. But there is nothing like that observed in RNA. So they haven't been able to show any sequence preference in RNA. So please pay attention to this because this is something that I used for my modeling. The position of the cytosine at -5 position and a thymine at +2 position. As you can see, they also listed the possible cleavage site because they tried to change the bases and then take this experiment. And we use these sequences for our modeling. So what happened is that the 5GVC model of the H-3 beta, it does not have any substrate, right? There is no nucleic acid in there. So how we can overcome this to run our simulation? So what we did is that in the E. coli topo 3, it has a crystal structure that has the DNA substrate bound to it. So the crystal structure is resolved as a complex of the H-3 beta and the DNA, and it shows the DNA in a close proximity. So there is no cleavage, but the DNA is in a close proximity. So what we did is that we superimposed our 5GVC, the H-3 beta, on that one. We obtained the 3D positions, the 3D coordinates of the nucleic acid, and we removed the E. coli topoisomerase 3. Okay, So we were left with the positions, 3D coordinates of our intended enzyme, the stop 3 beta, and the nucleic acid obtained from 205C, right? So now we have a complex, a working complex. But the thing is, the 205C was resolved with different DNA substrate. So the oligo was different. It's an 8-base oligo, but it's something that we don't really want. We want it to closely resemble this, where there is cytosine in -5 position and a thymine in +2 position. So you can change it. You can use computational tools to change those bases, and that's exactly what we did. We generated a DNA sequence that has a cytosine in -5 position and thymine in +2 position, and this is what it looks like. So there are 6 bases towards the 5' end. There is a probable cleavage site, and then there's 2 bases towards the 3' end. And you can change this DNA to RNA, which is pretty simple. We did just change the thymine to uracil for 2 of the models, that's the RNA3. And for RNA, we made 2 more models, which we copied from this list. So this list is really big and it's not exhaustive, right? So there can be more places where the topoisomerase can cut, and we cannot run so many models. So we just had to decide to take 2 out of there and make our model. So now we have one, let's call this a non-covalent model, and then we have 1 non-covalent DNA model and 3 non-covalent RNA models. One of them looks exactly like the DNA, it's just the thymine is replaced by uracil. So fine, we have 4 models, but we want to also check if there is any activity change when the covalent bond is formed. So this is where Dr. Chapagan's expertise was used to make this covalent model, where the tyrosine is, we can computationally make a patch where the tyrosine is bound to the DNA. Okay, so the tyrosine will bind to the 5' end of these 2 bases, and then there will be 6 bases left. So this is how the initial modeling was done. We have 8 models. So 1 DNA, covalent and non-covalent, and 3 RNAs, covalent and non-covalent, right? So the systems are generated through CharmGUI. This step is pretty simple. We have our models, we have the complex, and you can write it in one single PDB file. It's uploaded to CharmGUI. You can generate a water box through CharmGUI of the non-covalent models. The charm is the force field that we have used. Charm 36M is the force field that we used. It has a water box around it. This is the explicit solvent. We also have a 0.5 molar sodium chloride ions in there, and the temperature is 303 1.15 Kelvin to replicate the physiological system. The systems are equilibrated and minimized and equilibrated, minimized for 10,000 steps and then equilibrated. And then we run the production runs. So for the non-covalent, the production runs are pretty simple. You have your system and you can just run it through, we ran it through NAMD. We use the NAMD to run our simulations, and you get a trajectory file like this, where everything moves a little bit, right? In the molecular simulation, the enzyme and the protein moves a little bit, and you can get your necessary data of interactions between the residues and the nucleic acid from this one. Now, this is pretty simple, right? So you have everything, you can just upload it, but what about the covalent bonds? Because covalent bonds, we cannot generate them in MD simulations, and we don't have the crystal structure. So what do? So what we have is this. So in a previous paper, Dr. Chapagin and his colleagues, they were able to generate this file where the active tyrosine can be patched with the strand of the DNA. And we had to make a topology file and parameter files with the force field CHARM36M. And then this patch was applied through VMD to generate the PDB and the PSF files. So these files are necessary for running the simulation. So we have to moderate it a little bit for the human topoisomerase 3 beta, because this was made for an E. coli top 1, which is almost the same mechanistically. But we had to manipulate it and make it work for ours because ours is a shorter DNA. The enzyme is also different, and we had to do it for RNA also. So what we have to do is like we kind of have to strip all the, so you make the system in charm GUI, but you strip the water and the nucleic acid and the enzyme and the ions, everything separately. You apply the patch and then put them all together and run the simulation just the same. These simulations were run 3 times, so 3 replicas, and to like get the data and average them, so that we can get like not all the data is not too separate from each other. So they have to be similar and they have to be run 3 times to get the data. So first up, we try to understand the interaction, just a general interaction between the residues and the nucleic acid. So what we see is the cytosine at -2 position, it shows a pretty strong interaction with the arginine 194 and arginine 524 of the DNA. I'm going to show the exact locations of these. So we saw that, okay, this is like the bigger the number, the stronger the interaction. These are hydrogen bond occupancy numbers, and they show the interaction between two, let's say, two entities. Okay, so maybe two residues, or like a residue and ligand, or a residue and a nucleic acid. And our distance data showed that although in the noncovalent, the arginine and the cytosine can be a bit further away from each other. At the beginning, in the noncovalent, it is further away from each other, but in the covalent models, it's pretty close by. So the models are stable. So although
Yes,
can be further, yes.
a small question.
Yes.
About the hydrogen
Mhm.
occupancy numbers. I was reading the paper and
Yeah.
someone from classical machine learning, computer
Mhm.
science. I was wondering what actually that represents, but then you're pointing out that they are representing interactions like ligand and the molecule, the binding.
Mhm.
But that interaction is, at least that one is not abstract, like it is visualizable. It is easy to visualize.
Yes, I'll show it. And
Okay.
okay, so why don't we move on to that? This is just to show which residues interact with each other, and then when
It's
the
all
models are
right.
stable, the models do become stable after some time, and they're pretty close to each other, so the DNA doesn't float away. That's what I'm
All
trying to
right.
say in this page.
That's
And it's
okay. That's great.
okay.
Mm.
So why don't we go to the interactions? Okay, so you can put some cutoff. What the cutoffs you put is that you put a cutoff of 3.5 angstrom. The residues have to stay within 3.5 angstrom of each other. There is an angle. We usually put 30 degree. That one is a bit more like hydrogen bond always, all the bonds, hydrogen bond is always under like 4 angstrom, but we are also more, we do it more stringently. We put a 3.5 angstrom cutoff. The angle is a bit more difficult, but we use 30 degrees. Okay. So let's say during the whole run of the simulations, if two atoms are always under 3.5 angstrom and 30 degrees, so under this cutoff, they are close to each other, this will be given as a hydrogen bond. Okay. Now, during the run of the simulation, how long do we see that? Okay. So if in 100 nanosecond simulation, we see them together within 3.5 angstrom and 30 degrees for 90 nanoseconds, it's a 90%. Okay. And that means they're always very close to each other, that they're forming the hydrogen bond. So this is kind of the visual representation. I don't have a movie for this one because they take too long. Like if you run a movie, it takes too long. That talk gets like too long. But here, the arginine 194 forms very strong interaction with the base of the cytosine. Okay, so it can form 2 bonds, not just one. That's why the number can be more than 100%. If you take unique bonds, let's just say this atom and this atom, it will be less than 100. This atom and this atom, it will be less than 100. But if you add them together, sometimes it can be more than 100. Okay. So yes, the hydrogen bond is something that you can put the parameters for, and if an interaction falls under that parameter, you can say there is a hydrogen bond and the strength of that hydrogen bond. So
Um,
yes.
About the the type of bonds, the covalent
Mhm.
and non-covalent bonds.
Yeah.
It looks like I'm seeing this residue. I don't know whether it's a residue, an R194.
Yes,
Uh,
that's a residue.
okay. And the binding in the covalent model looks like it is closer. Like
So these interactions are not covalent or non-covalent. So the models are covalent and non-covalent. During a mu simulation, there is no covalent bond formation. A covalent bond cannot form during a mu simulation.
okay.
We can only see hydrogen bonds and hydrophobic bonds and some electrostatic bonds, and that's it, structural interactions. Okay. So the models are covalent and non-covalent. What we wanted to see if there is any differences between the interaction between the DNA and the enzyme after we have patched the covalent bond in. Okay. So we run it without any patching and without patching and try and see if there is anything going on like that sets them apart. So these are screenshots that I have taken during the whole simulation just to show how many residues usually interact. And the arginine 524 is pretty far away from the nucleic acid, is just because I took the screenshot at that time because I wanted to show actually I wanted to show the arginine 194 is close to the nucleic acid. So, and they do fluctuate. Okay. So you have to take the whole thing into consideration. Once it stabilizes, then you will start your measurement.
So what would be the greatest or the biggest takeaway you can give from these interactions, the covalent and the models, not the interactions, but the models.
Well, I wanted to give it a little later in the talk, but here we can find the residues that interact. So for first thing, we wanted to find the residues that interact and why the cytosine is preferred in -5 position. So what we suggest is that the cytosine interacts with the arginine 194. That's why it prefers the cytosine at -5 position. Now, the thing is, we did change the cytosine to guanine, but we also saw pretty good interaction. So it has been shown before that there is like only a cytosine can fit usually in E. coli topo 3, E. coli topo 1. So that's why E. coli topo 1 prefers a cytosine in -4 position, but we weren't able to show any kind of thing for this one. But still, we think that the arginine 194 has preference for the cytosine. That's why a cytosine base is preferred in that position as opposed to another like a guanine or adenine family.
Okay.
So technically, what we wanted to show is that which residues interact. This is very important because, as you can understand, this has not been shown in a crystal model. So whatever I'm finding out is the first time. And we want to see which of the residues interact with which bases. So we found that the arginine 524 and this glutamine and the lysine, they interact with the DNA backbone. And arginine 524 interacts with the cytosine 2 at -5 position, while also we saw this histidine that interacts with the thymine at -1 position. So right before the cleavage, like before the covalent bond that we made. So we think that these residues play an important role in the binding of the nucleic acid. So this is the first thing that we were able to observe. And other thing we are able to observe is some of the residues are very much fixed. So what happens is that when we put the magnesium ion, and then these residues show very little flexibility because they form like a complex, like a transient complex with the magnesium in between. And these residues are very important for topoisomerases because if they are mutated or deleted, topoisomerases lose their activity. So all in all, our finding, or actually what we wanted to see, if our model can replicate the physiological system, like if the topoisomerase is behaving as it should, okay? And we thought at this point, yes, we could replicate because we expected to see some strong interactions between the backbone and the bases with of the DNA with the residues of the enzyme, and we were able to kind of replicate this DXB motif with the magnesium in between, where also this lysine 10 is also important. If this is mutated, the topoisomerase 3 beta loses its activity, and we were able to see this activity of the complex formation, and which was also stable. And this is the flexibility. So if something is pretty rigid, it will have a low flexibility score, RMSF, and it held the whole for the whole simulation. It maintained its rigidity. So we think our model is pretty good for just for the DNA, and we wanted to see if we can find something different for RNA, like if the RNA is any different or is it the same, right? So for RNA, we had 3 models and actually 6, so covalent and non-covalent. And then we ran the simulations and we kind of see the same thing for the flexibility. All the same residues, the E9, D117, the lysine 10, and D119. They have the same rigidity with the magnesium in between, as you can see from this chart on the left. And the hydrogen bond analysis also do not highlight anything. So there was a lot more variable than we hoped or we expected. So simulations can go in different ways. Sometimes they do not replicate the same results. So this is challenge, and that's why you want to run 3 times, 5 times. For example, arginine 194, we thought are going to show strong interactions, but they don't always do that. For cytosine, it didn't show as a strong interaction as the DNA. For guanine, it showed weak interaction at some point, but a strong interaction in another model. So there are variabilities, and because of these variabilities, you want to take the whole thing together. So you want to see, like, when you put all the data together, do you see anything stand up? And nonetheless, we saw that the arginine 524 and there is another aspartic and in 520 residue, then the lysine 10, they interact with the backbone, and some other residues that interact with the bases. But this one stood up, the aspartic acid at 185. What we understood from these backbone residues is that they are part of a helix called a helix O, and this helix kind of moves a little bit to accommodate the DNA. So if you see the whole simulation, the helix O is more observable because it kind of moves a little bit to accommodate the fluctuation of the DNA during the simulation. Now, what we wanted to show here is that although large conformational changes such as the gate opening and DNA passing, those are like 2 big changes for this kind of simulation, and we won't be able to show that. But we do see small conformational changes like helices and sheets moving a little bit and accommodating the DNA. Why we wanted to show it is because there has been observations from crystal structure where they know these helices kind of move to accommodate the DNA, but the movement is not very visible. But in our simulations, these are much more visible, and you can see how the helices move around the DNA to accommodate it. But as I was saying, you want to take all the data together and put them together to see what stands up. And the residue that most stood up was the aspartic acid at 185 position. So this is pretty unique because RNA has 2' OH, right? And DNA do not. And this D85 was able to make an interaction with the 2' OH of the RNA. And this is something we do not observe in the DNA. Although the arginine 194, whether it's a cytosine or guanine, it makes very strong interaction with the base at 5 position, and arginine 524 makes a very strong interaction with the DNA backbone. And we're pretty sure these play a very important role in the interaction. But the D185 was the most unique thing. It showed interaction in the RNA, but did not show interaction in the DNA. And when we do sequence alignment of different type 1 topoisomerases over different kingdoms and organisms, we can see that the D85, R94, R554, they are highly conserved and they are very important for the DNA because they are highly conserved. So histidine 60 is also interacts with the 3' end, like the shorter end of our models. And we think that all in all, the DNA 185 is highly conserved across the species, and this might play an important role in the N-terminal domain part of binding the RNA, because the C terminals are different, right? But they still bind to RNA, different types of type 1 topoisomerases. So there are residues that are in the N-terminal domain that may play a role in the RNA activity. Although human type 3 alpha also has that D185, so maybe we need more mutation, like point mutation studies and more wet lab studies to make sure that these results can be further confirmed with in vitro studies. So all in all, we were able to find the important residues that interact with the RNA and the DNA, and we were able to find one residue that is different between the DNA and RNA, and it may have an important role in the RNA binding of H3 beta. We also did energy profiling to see if any of the substrates are preferred more than the other. We tried to do PMF where you kind of pull away the DNA from the binding pocket, but the problem is the interaction is so strong. When you pull the DNA, it kind of morphs the protein itself. So we had to go with the MMGBSA, binding free energy study, which does a lot of estimation, and there are some variation between the data. But our energy profile showed that although all the substrates are preferred, they all have like pretty good negative energy. Mind you, this can be a little bit off because of the estimation in the MMGBSA system. They didn't show any unique preference, so kind of all of them kind of showed the same preference for binding. So this was for the absolute binding free energy study. When we submitted the paper, we did have some questions like how we can prove the stability of the models, and we were able to show the magnesium and the schisel phosphate, the oxygen of this schisel phosphate group. Schisel phosphate means where the covalent attack is going to take place, and the schisel phosphate, they always maintain a very close distance over all the runs in all the simulations, and they have like very close. So it means that in the covalent system, we can non-covalent. So so this studies from the non-covalent models, and we can expect the covalent attack to occur because they always remain very close and the system is stable. Something interesting that we observed, it is in the covalent models. So we did have some simulation where they removed the magnesium, and most of the runs did have them. All the replica runs had the magnesium, but there were 2 runs where we removed the magnesium. And as I have said, the magnesium is pretty important, but what we saw that when we patch the covalent model in, and if we remove the magnesium and we run the simulation, the domains separate from each other. In 2 separate runs, they separated from each other in like a very large conformational change. And the one figure in the bottom, it kind of depicts the gate opening mechanism without any biasing force. So we didn't put any force like SMD to bias it. It's just the removal of magnesium kind of showed a large conformational change, which we thought was very interesting. And we think the magnesium may have to play a role in the conformational changes along with the attack, the cleavage cycle, the attack to take place and the religation. It may also have some conformational roles, the structural roles. Finally, we saw that even in the covalent models, the DNA can move further away a bit from the catalytic tyrosine, which we do not expect to happen. We want it to stay very close, but it can sometimes deviate away. But we saw that even in the crystal structures, the DNA can be further away. So maybe the cleavage has already occurred and the DNA is moving away, or the cleavage will occur a little bit later and the DNA will get even closer to the catalytic tyrosine. So both of those can happen, and our data was kind of in agreement with the crystallographical findings, where we saw our DNA is in some of the runs, our DNA was a bit too further away from the catalytic cycle, from the catalytic tyrosine. So all in all, this is what we were able to find, and we were able to show like our models replicate crystal structures, and in lieu of a crystal structure, because crystal structures are hard to find, we can find important data, important residues, and important interactions with our models. So that is actually the finding of our work. Okay, so they can give us insights because we don't have crystal structures yet, and crystal structures are harder to make. I kid you not, we also make proteins in our lab. We send them to our collaborators. It can take years just to make the DNA stick to the protein and have that crystal structure and have that structure elucidated and get the data. Okay, so those things, they work in a different way. They just work on their own. So you get the crystal or you're not, but you just keep trying and trying, and it can take a long time. In the meantime, uh, if someone wants to use my models for further study, they can totally use it for whatever they need. And we can see a small conformational changes, although large conformational changes are rare to see. Although we did see one with the removal of the magnesium, but yeah, we are more prone to, we can more easily see the small conformational changes. And we have used these models. We ourselves have used this model for molecular docking and screening for computer-aided drug design. You can use the non-covalent models, so the DNA and the protein, and then you can extract different time frames. And then if you have a computational mod like a drug library, you can try and dock those drugs in those pockets, and then you can like do further in vitro assays with those. So these were my findings, kind of. And I would like to acknowledge my PIs, both my PIs, Dr. Ben Chapagin and Dr. Yuqing Shirin. Dr. Yuqing Shirin is a pioneer in topoisomerase studies in this country, and her lab focuses mostly on type 1 topoisomerases, mycobacteria, and E. coli. Mycobacteria owns only one type 1 topoisomerase, making it a very important drug target to tackle TB. And Dr. Chapagin expertise on molecular modeling. So his lab has models on bacterial membrane, lentipeptides that can work as bacteria, a lot of membrane studies of drugs passing through those membrane. And then for my project, he focuses on the topoisomerases and their modelings. I'll also acknowledge my dissertation year follow-up provided kindly by FIU EGS. And this work was also done in part with the NIH grant. And thank you for listening. I hope you guys enjoyed it.
Thank you, Yasser. And I want also to thank your team, Dr. Chapagani, Dr. Ching, and Shamita. Really, we really appreciate. The first question from me is about, you actually talked about it. If someone wanted to pick up this work and they like just to replicate what you've done, and maybe they can start from there and extend it. I was looking at the paper to look at the resources, and there was kind of not direct how to access them, but maybe you'll highlight on that. Then another question is about the magnesium ion. I've seen in we have seen in the interactions. I get the feeling that the magnesium ion enhances the interactions between the residues. And somewhere it was removed, and I didn't see much of a big change in the interactions. Maybe I missed that.
No, okay. So first thing, if you want the resources, I think in the paper we have the Zenodo is a database where you can go and download the PDB and PSF files along with the DCD files.
Zenodo, okay. Okay, I'll look that up, but I'm seeing like
Yeah.
rcsb.org and
No, RCSB is the PDB. So you can just get the structure there.
okay.
But I think in the end, it is in the supplemental files. You have to go into the supplemental files and then see, and then you have to see that where they are deposited. Okay.
Sure.
Okay, for the magnesium one, we never really did residue. I didn't show the residue interaction with magnesium. So the magnesium plays an important role in the cleavage because it forms before the covalent attack takes place. Okay, so let's go here. There is covalent attack has to take place. Okay, for the covalent bond to form, the nucleophilic attack has to take place for the bond to form. Here, there is always a magnesium. So these figures, they don't have the magnesium, unfortunately. When the magnesium is there, it forms a nice complex, a transient complex to stabilize the whole thing, and the attack takes place. Okay. And once it's time to religate, again, it forms a bond with the nearby residues, the aspartic acids and the OH and the oxygen of the DNA backbone. And it again forms a transient complex, and then the DNA religation attack takes place. Okay. If you remove this magnesium, those complexes won't take place, and the catalytic cycle won't be complete. So this is the importance of the magnesium. In test tubes, in vitro experiments, you can put a lot of EDTA to get rid of the magnesium, and then you'll see that the experiments do not work anymore, the relaxation experiments. I wish, because we are already running out of time, I cannot show you the gels and the things that you can do in the lab. But you're a machine learning guy, so I don't know if you would enjoy them. But yeah, I could show you like the actual impact of those things, doing those things.
Okay. The reason why I was asking is I've seen some papers where they are trying to predict some sort of binding affinity, and that is from machine learning. And within the process of looking at these molecules, the ligand and the molecule, they add some decoys, some sort of decoys.
Okay.
And I was, so it came to my mind as like, is magnesium acting as a decoy? But that was the distraction. And
so at this point, I guess if you want to say a ligand, I think the DNA, although it is pretty big, it has 8 bases, but it's the ligand, it's the substrate, okay, for this enzyme. The magnesium is just it's something else. It's not the ligand. It plays an important role in stabilizing the whole thing, but it's not the ligand, because, well, how can I say, the enzyme doesn't really act on the magnesium. The enzyme acts on the DNA. So I didn't show any interaction because these systems, they fall apart. Without the magnesium, the covalent models, they fall apart. The non-covalent models, so we have run models without the magnesium for the non-covalent models, but they don't really show any unusual activity. But interestingly, the covalent models where we patched a bond between the DNA and the enzyme, the removal of magnesium separates the domains. So the system fails. That's why we didn't do any further study. But it's just an observation. Like without the magnesium, is there any structural changes at a lytic cycle? It's something just we wanted to I just wanted to share with you. It's something that you can see. So I hope like you enjoyed the talk, because I guess you are more machine learning, but this is structural biochemistry, biophysics. So
yes, members, if anyone has a question, just unmute yourself as usual. I'm also curious about these residues, especially I'm seeing cytosine, and it's it tends to be performing better in the interaction. And my question is, are there residues that you can say these are the residues that are playing the most crucial role than others?
So yes, in case of DNA, we are thinking that the arginine 194, the residue. So when you say cytosine, it would be either the nucleotide or the base. So cytosine is the base or the nucleotide of the DNA, and it shows pretty strong interaction with the arginine 194, and it has been shown in different other type 1 topoisomerases like E. coli. There are some arginine. So as you can understand, the tyrosine is the most important residue, right? If you remove this, the enzyme won't work at all. This is the catalytic tyrosine that causes the DNA breakage. But there has been studies of residues that are farther away. So let's say if this is the catalytic side, farther away down, the residues that are farther away, they can also play an important role in the activity. If you either mutate them or get rid of them, the DNA will lose its activity completely or a lot. It just won't work that well. So we think the arginine 524 and arginine 194, these residues may play that role. If you remove them or mutate them, the DNA may lose its activity because they are conserved across species. So this alignment is a conservation study, and this shows if some residues are conserved, they are more important. It's just how we kind of understand the data.
Okay, lastly for me is about like the machine learning person. I'm thinking if what could be the biggest impediment like for this work to be transformed into machine learning models or language models. You talked about some hard thing to get like data. I think you were talking about some component of the DNA. But if it is plausible, can we model these interactions using maybe language models or some other deep learning architectures?
Well, I think right now it's gotten easier. It's like nothing is harder. So you see how I had to make these models, like because there is no crystal structure with the DNA and the enzyme, I had to get another enzyme and then stamp it and then take the nucleic acid. You don't have to do this anymore. You go to AlphaFold 3 website, you just put a DNA sequence and I put the enzyme sequence, and ta-da, you have everything. So right now things are getting easier. Only thing I guess would be harder is this thing, the covalent bond formation. So if you go to PDB, there is like a lot less enzyme nucleotide complex as opposed to just enzymes. And I guess this is something that you can address. So if you want to do machine learning, something like AlphaFold 3 or I don't know, Rosetta Fold or OpenFold, so many things are out there, right? And you can use them to maybe make better complexes, because the complexes are not always well interpreted through machine learning, because the machine learning learns from these and they have a short, either nothing or short DNAs. So if you can work on longer DNAs or DNA that are already has formed a covalent complex with the enzyme, those are much harder to address. And these take a lot more work because you have to write scripts and you have to make sure every step of the way that what you did is correct. So those are the more challenging.
All right. Thank you very much for this great work and the presentation. I guess there are no more questions. We really want to thank you once again for creating time and really present this work to us.
I hope I interested in molecular dynamics and some of the like actually structural study.
Absolutely. People were really enthusiastic about this paper. So thank you very much. We will be uploading this this video to our YouTube channel. And yeah, to the rest of the members, thank you for attending, and we will meet in our next session. I think bye-bye to everyone, and bye-bye to Yasil and the team.
Okay.
Thank you. Bye-bye.
Bye-bye.
Thank you. Bye.
You're welcome.
Thank you. Bye-bye.
Bye-bye.